Today's topic is about the histology of the nerves and about its classification as well. Within this, I will be looking at how the nerves can be classified, and also will be looking at the major most abundant cell that is found in the central nervous system, which is the neuroglia. Now, if I look at the neuroglia, there are four types that you can see. This includes astrocytes, oligodendrocytes, microglia, as well as the ependymal cells. The neuroglia usually has its origin from either the neural crest cells or the neural tube cells. But this is not always true. If a statement arises where it is said that all the neuroglia are rising from the neural crest cells or the neural tube cells, or in fact neural progenitor cells, it is not true because of the fact of microglia. Microglia has a bone marrow origin, which means that it is a differentiated cell that comes from monocyte, similar to that of cells that are found in the lungs, which has the capacity to carry out immune functions, and also the Kupffer cells that are found in the liver. So, in addition to the neuroglia, now, another statement that we usually come across is that this is the most abundant type of cell that you see in the central nervous system. In fact, it is around 10 times to that of the neurons. And the main function carried out by the neuroglia is the provision of support for the functioning of neurons. It maintains a friendly microenvironment for the better functioning of the neurons. Now, the intercellular network that you see between the neurons and the neuroglia, which resembles a sort of a fibrous network, is known as the neuropile. So, neuropile is the resembled fibrous intercellular network that is composed of both neuroglia as well as the neurons. Now, if I go further into the topic, oligodendrocytes, again a very abundant cell, not the most, because astrocytes is the most most cell that is found in the highest quantity, but the second most abundant cell, which can be easily identified because of the presence of a dense nuclei at its center. And also something very specific about oligodendrocyte is the presence of a clear cytoplasm. Now, unlike to that of the other cells, especially microglia, which is heavily stained by H&E stains, however, oligodendrocytes are not so. The other function that it carries out is the ability to form myelin sheets for the axons. Something very specific about oligodendrocytes is that it can form myelin for multiple axons rather than that of the shown, which usually involves in myelination of a single axon only. Now, astrocytes are the most abundant type of cells, most numerous. There are two types of astrocytes that you come across. One would be the protoplasmic, which is a branched short cell that is present mostly in the gray matter. The other type of the astrocytes is known as fibrous astrocyte, which is usually found within the white matter. Now, this is associated with the specific protein that is known as glial fibril acid protein. The common abbreviation that we use is GFAP. This protein is an important marker for tumors that are arising from the astrocytes. Now, most of you know that a very common neural tumor that we come across in our clinical practice is the astrocytoma. Now, once the astrocytoma is uh, found within an individual, to identify as well as to evaluate the growth of the astrocytes, we can do GFAP levels. Now that is something very significant and important in our clinical practice. Therefore, should remember this. And some of the functions that are carried out. As I said, these neuroglia provides a support to the action of the neurons. For this support, there should be the maintenance of homeostasis, a constant internal environment. This is maintained by the action of neurons. Now, neurons, the cytoplasm, are involved in multiple number of transport channels that are present within the membranes, which are involved in homeostasis, particularly maintenance of the electrolytic levels, intercellularly as well as intracellularly. Now, the maintenance, again, the most special consideration that it does is for the potassium homeostasis. In addition to the buffering action of the electrolytes, this also carries out uh, important functions which are related with differentiation of neurons, especially in the embryological state, where neural crystals, neurons, has to be differentiated and undergo modifications to perform the functions of an adult human being. For this, it plays important functions. And in addition to this, it also plays a role in angiogenesis, vasodilation, vasodilation, where the vessels that are found in proximity to the neurons are allowed to dilate and provide more nourishment, more blood to the specific area. And also, there are extending processes which are coming out from the astrocytes that plays a key role in the neuroplasticity. Neuroplasticity. So these are few functions that you would see in the the activity of astrocytes. Something that I have to say is that these astrocytes sometimes are found as a lining to the meninges. Now, if there's the meninges, there's this lining of the astrocytes which performs the function as a glial limiting membrane. Now there's the ependymal cells. Ependymal cells are usually found as cuboidal or columnar and are found within the ventricles as well as the central canal. Now, the most significance of the presence of it in these ventricles is the fact that it is involved in movement of cerebrospinal fluid as well as involved in absorption, which will prevent the occurrence of increased central increased cerebral pressure which is quite an important topic when it comes to physiology. Just know that it plays its role in the regulation of the CSF content in the brain. And then there's the microglia. As I said, microglia are very less in quantity, but the important factor is that it plays its role in the immunological function. Now, it has its actions on the defense against foreign agents, as well as it sometimes act as a antigen presenting cells. Sometimes the antigen presenting cells causes the production of certain antibodies, which will lead to destruction of the myelin sheaths that are present inside the central nervous system of an individual. This entity of the disease is known as multiple sclerosis. So again, in within the pathophysiology of multiple sclerosis, microglia is somewhat an important entity. Addition to this, something very significant about this is that since this is a cell that is arising from the monocytes, so it has its progenitor cells within the bone marrow, and the cytoplasm of this cell is heavily stained by H and E. Now, in addition to neuroglia, I will be looking at the Schwann cells. Schwann cells are found within the peripheral nervous system, is involved in myelination of the nerves. Now, as I said, most of the time what you would see is that the Schwann cells are involved in myelination of an axon, and a single Schwann cell will have its function only on enwrapping a single axon only, not more than that. But this is not true when it comes to oligodendrocytes. Oligodendrocytes will have the capacity to uh, enroll more number of or multiple number of axons. The Schwann cells, now initially what happens is that the Schwann cell starts wrapping around the axon or the axolemma, and with time the membrane will fold in multiple wounds in a very consecutive way, in a very concentric way, and by doing that, later these membranes are very abundant of proteins as well as lipids. The specialized lipid that is present is known as the myelin. With the enwrapping, it forms a sheath-like structure that provides insulation to the axon and prevents the electrical energy from dissipating and becoming short-circuited once the flow of the current takes place. Now, this is known as the myelin sheath. Something that I can see by looking at this structure is that the cytoplasm and the cellular contents are usually set out to the periphery. It is taken out, but this is a living layer which requires its nourishment as well. So during the process where it requires uh, the movement of material, it allows a small quantity of cytoplasm to pass in between these layers, carrying both oxygen as well as nourishment. Right. So these Schwann cells are found within the axon, and in between sometimes there are non-insulated regions which are known as, these are nodal gaps which are known as nodes of Ranvier. Within the nodes of Ranvier you find the most concentrated levels of sodium and potassium channels, allowing electrode or allowing ion transitions to take place for the depolarization purpose. Now, usually when the current has to flow within an axon, what would happen is that the depolarization only happens at the nodes of Ranvier, and it tends to jump from one node to another in a conductive modality known as saltatory path. By doing this, it makes sure that the depolarization or the action potential not to be generated at each and every point of the axon, and it since the current can jump from one place to another, what would happen is that the rate at which the electrical current is or the nerve stimulation is conducted is extremely enhanced. The better or the larger the Schwann cells, the lesser the nodes of Ranvier. When the nodes of Ranvier are lesser, what would happen is that the conduction rate will be increased as well. Once this is of course a clinical point. Once a nerve of the peripheral nervous system becomes disrupted or torn or underwent some sort of a damage, usually the neurons does not have the capacity to produce new cells. It is not, it does not have the ability to regenerate. But once this is broken, again the nerve can be realigned as well as the conduction can be re-established by the action of the Schwann cells. The Schwann cell you sometimes wraps around the point at which there was this disruption and allows re-establishment of the electrical pathway. So it plays its important function in battling out neuropraxia. Right. Before going to the question, another as I said that I was also interested on to relate about the classification of the nervous system. Now the nervous system consists of mixed nerves and sensory nerves only. Now what you would see is that the mixed nerves are divided into three types, particularly according to the nerve fiber that you come across: A, B and C. A is further divided into alpha, beta, gamma and delta. Alpha are completely motor neurons. These are large, heavily myelinated and involved in fast conduction of nervous stimulations. The beta is a sensory nerve, mediumly myelinated and is involved in conducting sensation of touch and pressure. Gamma also in a similar manner, but this is a motor fiber. But this composed of a sensory fiber which are involved in. I'm sorry. Gamma is a motor fiber which is involved in innervating intrafusal fibers of a muscle. Mostly important in maintenance of the tone. As I said, there was this alpha motor innervation as well as gamma motor innervation. Gamma motor innervations is involved in the maintenance of the tone. Again, a medium to heavily myelinated nerve fiber. Then there's the delta. Delta again important in touch, pressure, temperature, and fast pain. Now there are two types of pain which I am not going to elaborate in this video, but there's the fast pain and the slow pain, slow dull type of pain as well as the fast pain. In a pricking injury you would sense the fast pain, whereas in an abdominal pain mostly it is the slow pain that causes this sensation. B. Now delta is not does not contains that much of myelin, and it is also involved in moderate to slow rate of conduction velocity when it comes to uh, nerve stimulation conductions. Right. The B type. B type is found in the, if you can remember, in the autonomic nerves, which compose of parasympathetic and sympathetic. There was the pre-ganglionic nerve and there was the post-ganglionic nerve, which causes action of the effector. And this is the ganglion. This was the pre-ganglionic neuron, and this is the post-ganglionic neuron. The pre-ganglionic autonomic fiber is composed of the B type. Again, not well myelinated and has a moderate rate of conduction. Then there's the C type. C type, as you can remember, it carries a sensation of slow pain and also found post-ganglionic nerve fibers. These are preganglionic B type, and in the postganglionic nerve fibers of the autonomic is composed of C type of mixed nerves. Good. So this composed of both sensory and motor. Right. Then the sensory nerves are classified as well. Now they are classified, they are given a number 1, 2, 3, and 4. One has 1A as well as 1B. A is an fiber which is involved in muscle spindle innervation. Plays an important role in pressure that conducting sensations of the pressure to the muscle spindle as well as regulating its function. Now B type is found in the Golgi tendon, again within the muscle. These are large and myelinated. Then there's type 2. Type 2 involved in moderate conduction of nerve stimulation, mediumly myelinated, and these are usually found in carrying out sensation of touch and pressure. Third type, again touch and pressure, because there are multiple number of receptor units which are involved in carrying out sensation of these two modalities. That's why there are different types. And also fast pain and temperature. Now these are the same types that were initially thought as A, B, and C. However, the sensory fibers are further classified by giving their number according to the myelination as well as the rate of conduction that you can see in these nerves. So touch, pressure, fast pain, and temperature. Number 4, 4-type sensory nerves, which are the slowest and found unmyelinated, are found carrying sensory information of pain, temperature, slow pain, and olfaction. So the smell. Now what happens from A to B to C is the myelination usually reduces. Myelination reduces. Rate of conduction usually reduces. Right. So we'll go to the questions. In an unmyelinated nerve, conduction is saltatory. No, for the saltatory conduction to take place, there should be nodes of Ranvier, which is found only in the nodal gaps that are found in between the Schwann cells. So this is a false statement. Action potential is generated in the cell body. Again, a wrong statement, because the action potential is usually is initiated at the axon hillock, or the initiation of the axon. So not the cell body. Axon hillock is the terminal part of a cell body. So this is bit controversial actually, but if the exact location that has to be specified here would be axon hillock, which has more potential than saying that it's the cell body. So the question is bit deranged. I'm not sure about the question, the answer. Myelin sheath of an axon is formed by several Schwann cells. Yes, it is formed by number of Schwann cells, but because a single Schwann cell has the capacity to enwrap only a single axon. Myelin is absent in ganglion nodes. Yes, that's a true statement. In the central nervous system, the myelin sheath is formed by oligodendrocytes. Yes, that's true. So this is it. uh, Here I have tried to bring about most of the statements, most of the questions, and as well as the most examined points of histology of the nerves. um, in the next lesson I will be looking at a more clinical perspective of the nerves, and how it correlates with the disease pathology as well. So until next time, thank you.